Hello, everyone. This recording deals with the part of Chapter 4 in our textbook in which we talk about carbohydrates. So first of all, what are carbohydrates? Well, you should read this portion of Chapter 4 in our textbook to learn all about carbohydrates. So their name sounds like hydrated carbon or carbon with water. That's not really what they look like chemically, but they do consist of carbon, hydrogen, and oxygen. And because of that, we sometimes abbreviate them CHO for carbon, hydrogen, and oxygen. So there is a handout that I gave out in class that had a flow chart of all of the carbohydrates, and that flow chart is in this slideshow, so I will point it out to it when we get there. So how do we classify carbohydrates? We have simple carbohydrates, or sometimes you could say simple sugars or just sugar. So if you say the word sugar, you're usually referring to a simple carbohydrate, either a monosaccharide or a disaccharide. So the monosaccharide consists of a single ring of atoms, whereas a disaccharide consists of two rings that are linked together. We also have complex carbohydrates, and those could be oligosaccharides where we have more than two, we have a few rings linked together, or polysaccharides where we have many rings linked together. So let's start with the monosaccharides. So the monosaccharides that are important in our diets are glucose, fructose, and galactose. And these pictures right here are from our textbook. So glucose is the most abundant sugar molecule in our diet. We don't typically eat it by itself. I hope that will become clear as we continue through the talk. Fruit sugar is found alone. It's found naturally in fruits, vegetables, and honey. Galactose does not occur alone in foods. It actually forms chemical bonds with glucose and other sugars to form either disaccharides like lactose or oligosaccharides. So, and what I wanted to briefly explain is that when we draw structures of sugars, we can draw them in a straight chain form. Now imagine that I take the straight chain form and I bend the carbons around like this. And I have an OH group right here and a carbon doubly bonded to an oxygen. So what happens is that a chemical reaction takes place in which the ring is closed. And another consequence of that reaction is this oxygen right here becomes another OH group. Now, depending on how that reaction takes place, this OH group at carbon number 1, where I closed the ring, could end up below the plane of this ring or could end up above. If the OH ends up below the plane of the ring, we call that alpha. And if the OH ends up above the plane of the ring, we call that beta. So it's at the point of the ring closure. So here for glucose, the ring closes at carbon number 1. So the other thing that we should point out is that glucose is being shown here. Galactose is not, but when glucose and galactose form rings that are 6-membered rings, then they're usually drawn as hexagons in textbooks. That's not really what they look like, but that's usually how they're drawn. We can also look at fructose. So fructose has a chemical structure that's slightly different, but it also has this C double bond O right here and an OH group. And again, if I take this ring and sort of bend it around, a similar chemical reaction will take place, and I will close the ring. And again, one of the consequences of this reaction is that you form another OH at the point of the ring closure. For fructose, we're going to close the ring at carbon number 2. But the OH is below the plane of the ring, and we use the same convention where we say that this is the alpha form of fructose because the OH ended up below the plane of the ring. So you might notice right here that fructose is different from glucose and galactose in that fructose forms a 5-membered ring. We usually draw it as a pentagon. Again, that's not exactly what it looks like, but that's how we draw it in the textbooks. So those are our important monosaccharides. Now let's talk about disaccharides that are important in the diet. So there's lactose. So lactose is found in dairy products. So we commonly call this as milk sugar. So if you have milk or any kind of milk product, it will have lactose in it. So milk, cheese, yogurt, ice cream, um, any kind of cream sauces for pasta or anything like that, all of them will have lactose. And lactose consists of glucose and galactose chemically bonded together. Another important disaccharide is sucrose. So sucrose is just table sugar. So sugar cane or sugar beets are sources of sucrose. And they consist of glucose and fructose bonded together. So let's look at how we form lactose. So lactose is made up of galactose and glucose. So over here on the left, I drew beta galactose. I drew its ring in the closed configuration. And notice that the OH is above the plane of the ring. So this is beta galactose. And here is alpha glucose, and it is in the alpha configuration. But what I'm going to do is I'm going to form a bond between this OH and the carbon that was the point of the ring closure, and then this OH over here. And what I'm going to do is one of the products is going to be water. So if I take off 2 hydrogens and 1 oxygen, that gives me my water, and it gives me a bond between these 2 monosaccharides, so 2 carbons with an oxygen in between. So we call this a beta bond because before I did the reaction, the OH that I started with was in the beta configuration on the galactose that's on the left. So we can call this a dehydration reaction, or maybe even a condensation reaction. So one monosaccharide donates an OH and the other one donates a hydrogen, and then that makes one of the products water. And a new chemical bond, we call this a beta bond, forms between the 2 monosaccharides. So this is the disaccharide lactose. It consists of 2 monosaccharides, a galactose and glucose. What about sucrose? Well, this is glucose and fructose bonded together. So I can draw it like this. I'm going to use the alpha configuration of glucose. So again, here's the point of the ring closure right here at this carbon. And you can see that the OH is below the plane of the ring, so we call it alpha. This is the beta fructose. Remember, it was a 5-membered ring. And I'm going to use the beta form of the fructose, where the OH is above the plane of the ring. And I'm going to form the bond between these 2 carbons. And again, one of the sugars donates an OH and one donates a hydrogen, so we get water and we get a bond between them. So our textbook authors called it an alpha bond. I don't think that's correct. If you look at the glucose, remember the OH before we did the chemical reaction, this was alpha and this is beta. So typically we just don't call this either an alpha or a beta bond. It's simply a bond between an alpha glucose and a beta fructose, and that gives us the disaccharide sucrose. So let's go on to complex carbohydrates. So we have oligosaccharides. So oligo means few. So the 3 examples I have here are raffinose, stachyose, and verbascose. So raffinose is found in beans, cabbage, Brussels sprouts, broccoli, and whole grains. Stachyose and verbascose are found in beans. So raffinose is a trisaccharide, so we have 3 structures, 3 rings, 3 monosaccharides. Stachyose is a tetrasaccharide, or 4 monosaccharides, and verbascose has 5. So this picture describes how we can build these compounds. So here's our sucrose right here. So the sucrose consisted of glucose and a fructose. And what we do next to make raffinose is we take a galactose. And here's the galactose. This would have been an OH before you did the chemical reaction, and the OH is below the plane of the ring. So that's an alpha bond involving galactose. And in fact, all of these are alpha bonds involving galactose. So sucrose is just a glucose and a fructose. Raffinose is glucose, fructose, and 1 galactose. Stachyose is glucose, fructose, and 2 galactoses. and verbascose is glucose, fructose, and 3 galactoses. What about polysaccharides? Well, the two different kinds of polysaccharides that I want to tell you about today are either digestible or not digestible. So the digestible carbohydrate that I want to talk to about you today is, you've probably heard of starch. It consists of two molecules that chemists would call amylose or amylopectin. Starch is digestible. It is made up of glucoses, just glucoses, and all of the glucoses are alpha. Cellulose is not digestible. It's made up of glucoses too, but here all of the glucoses are in the beta configuration. So let's look at some of the structures. So if I wanted to build the structure of a starch molecule, I'd start out with glucose. So here I've just drawn two glucoses, and we can see that both of them, again, here's the ring closure over here, and here's the ring closure over here. Both of these are alpha glucoses. I'm going to link them here by doing a dehydration or a condensation reaction like I showed you before. So since the point of the ring closure, the OH was in the alpha position, I'm going to call this an alpha bond. And I can link two of the glucoses together, or this picture shows 4 glucoses, but amylose can be thousands of glucoses long. And all of the glucoses that make up amylose are all the same. They're all glucoses and they're all alpha glucoses. So that means the bonds between them are referred to as alpha bonds. Well, amylopectin is similar. It has a long chain, as you can see down here, but what we do is we can actually create a branch. So amylopectin is a branch chain. So if we look at this picture that's in our textbook, each of these little green hexagons right here is meant to represent glucose. So amylose, like I just showed you, is a long chain, and amylopectin is a long chain with lots of branch points. And when we say the word starch, starch is a mixture of both amylose and amylopectin generally. And we find these in the kinds of plants, grains such as wheat, legumes, tubers like potatoes. So, so think of things like rice and pasta and bread and potatoes and things like that, where we have starch. And again, a chemist would call that amylose and amylopectin. And again, both of those molecules are all made up of one thing, and that's alpha glucose, and the bonds between them are called alpha bonds. Well, what about a non-digestible carbohydrate like cellulose? Let me show you how we put that together. So if I'm going to make cellulose, I'm not going to start with alpha this time. I'm going to start with beta glucose. So here are my glucose molecules, and there's the point of the ring closure, and the OH is above the plane of the ring. So these are both beta glucoses. Now I want to form a bond between this OH here and this OH here, but they're not next to each other, so that's not going to work. So I'm going to take this glucose and I'm going to turn it upside down. So if I turn it upside down, now the OHs are next to each other, and then I can do a reaction just like I showed you before. And now I have a bond between the two glucoses. So this is a beta bond because I started out with glucose that was in the beta conformation. And the way that this works is that you have one glucose is sort of right side up and another one is upside down. So when you make glucose, when you link, again, thousands of them together, you have right side up, upside down, right side up, upside down. So that gives cellulose a different structure than the starch. Remember, the starch consisted of amylose and amylopectin that was held together by alpha bonds. The cellulose is made up of beta bonds. So if you were in class today, you would have gotten this handout. So I like this handout because we look at simple carbohydrates and complex carbohydrates, and we can divide them into those two categories. The monosaccharides, just one ring, glucose, fructose, and galactose. The disaccharides that we mention in terms of our diet, or lactose and sucrose. Trehalose is not as important nutritionally, and I will talk about maltose in a second. Lactose, it consists of glucose and galactose, and it's the link is a beta bond. Sucrose, it consists of glucose and fructose, and we don't refer to the bond as either alpha or beta. The oligosaccharides, raffinose, stachyose, and verbascose, remember, consisted of glucose and fructose and galactose, with stachyose having one galactose, raffinose having one galactose, stachyose having two, and verbascose having three. Now over here, we have starch, and starch is amylose or amylopectin, and it's made of all alpha glucoses, so they have alpha bonds. I'll get to glycogen in a minute. Now this is dietary fiber. So dietary fiber, there are lots and lots of different molecules that we consider to be dietary fiber. So the cellulose that I just told you about, the cellulose, that's an example of just one of them. So now that we know something about the structure of the carbohydrates, we can figure out what happens to the food that we eat when we digest them. So remember, we learned in Chapter 3 that we have digestion, absorption, and elimination. They all occur in the gastrointestinal tract. Digestion is the process by which the food molecules are broken down to their components. Absorption means these products are taken up through the cells that line the small intestine. And elimination is the process by which undigested food and wastes are removed from the body. So here is an outline of carbohydrate digestion. So this shows the entire carbohydrate digestion in its entirety. I'm going to break this down with some figures from some different textbooks that show some of the details. But what we can say right now is that the primary goal of carbohydrate digestion is to break down polysaccharides, disaccharides into monosaccharides. Only monosaccharides, just that one ring, only monosaccharides can be absorbed by the enterocytes. Those are the cells that line the small intestine. Even 2 rings is too big. So everything has to be digested to a single ring, a single monosaccharide, before it can be absorbed. So let's look at that in detail. Okay, the other day we learned that digestion begins in the mouth. So when we eat our food, we chew it and we break it up. It's mechanically digested and mixed with saliva. And one of the ingredients of saliva is salivary amylase. So the digestion of carbohydrates begins in the mouth with the salivary amylase. So again, remember, the goal is that everything, all the carbohydrates we eat, from simple to complex, have to be broken down to monosaccharides to be absorbed. If they can't, we can't absorb them. So here's an example right here. So if we look at starch, remember, starch is a mixture of amylose and amylopectin. And remember, the amylose consisted of glucoses, all alpha glucoses, all with alpha bonds. And remember, the amylopectin was similar, except it had these branch points. So the saliva has salivary alpha amylase. Notice that all the sugars ended in ose and the enzymes end in ase. So it even says alpha amylase because the enzyme is going to catalyze the breaking of these bonds, and we call this hydrolysis because, remember, we took water away when we formed the bond. Now we're going to add the water back. So the food is not in the mouth for that long. So we have these long amylose and amylopectins in the starch that we eat, and you can see that they're broken down into smaller molecules. So we can call these smaller molecules dextrins, but they're still too big to be absorbed. When we swallow the food and we get it into the stomach, there is no further digestion. And the reason for that is that, remember, we talked about in Chapter 3 that the stomach contains hydrochloric acid, and that it was going to kill the enzyme. So the alpha amylase doesn't function when it encounters the hydrochloric acid. So we've killed our salivary amylase, and there's no further digestion in the stomach. So now we need to go on to the small intestine, where quite a lot happens. So remember I showed you this before, that we have these circular folds of the small intestine, and they're very highly folded. And if you look at this little box right here to blow this up, the folds have folds on them. These are called villi. So these are these finger-like projections. And if we blow it up even more, we can see that these are lined with cells, and we call them enterocytes. And then these have little microvilli. And you can only really see them under the microscope right here. So we have to get our digestion products inside these enterocytes. So again, these are the cells that line the small intestine. And there are different sort of faces of the cell. So these folds right here, the microvilli, they face the gut where our food is being digested. And so that's called the apical membrane right here. And this is the basolateral membrane down here. And that's going to be connected to the circulatory system that will bring absorbed carbohydrates to the portal vein and to the liver. Okay, so let's look at see what happens there. So again, the small intestine, that's the major site for nutrient digestion and absorption, and especially true for carbohydrates. So remember, we had from the mouth, we had taken our amylose and our amylopectin, and we had broken them up into smaller pieces. The oligosaccharides and the disaccharides were not digested at all in the mouth. So what we need to do here is that again, all of our carbohydrates need to be digested to monosaccharides. Even something like a disaccharide is too big. And the monosaccharides are absorbed. They pass from the lumen of the small intestine into the enterocytes. Those are the cells that line the small intestine. So I showed you this figure before too. So again, our chyme arrives from the stomach to the small intestine. And now we need another sort of batch of amylase. Remember, we had salivary amylase, but it was killed in the stomach. So now we have a different source of amylase, and that comes from the pancreas. So that's pancreatic amylase. So here's our pancreas right here. So the pancreas is going to secrete sodium bicarbonate. So it's going to bring the pH up to neutral. Remember, the stomach's very acidic. But we also have these acinar cells that secrete digestive enzymes. And what's important to our discussion now is the pancreatic amylase. So if we look at what happens now, here we are in the small intestine. There is our pancreas right here. And there's the pancreatic duct that secretes the pancreatic alpha amylase into the small intestine. And now we can take those smaller pieces, those dextrins from the mouth, and break them down even smaller. So now we've broken them down into maltose. And maltose is just simply two alpha glucoses linked together with an alpha bond. If we do the same thing with our amylopectin, we will also get some small molecules here, but remember, there are branch points here, so some of these little molecules will have branch points. So the pancreatic amylase will break most of those alpha bonds in amylose and amylopectin, but not the branches. And we need additional help because, again, we're still not small enough for the enterocytes to be able to absorb those digestion products. So what do we need? Well, we need, here's our maltose. We need maltase to break the bond between these two glucoses, so we now have individual glucoses. And we have some maltose here from amylopectin digestion, and maltase will help us here as well. We also have, you can call this isomaltase if you like, and that is the enzyme that is able to hydrolyze that branch point. So now, what started out as starch, amylose and amylopectin, now we have a whole pile of alpha glucose. But if we've had a mixed carbohydrate meal, maybe we had something with a milk product in it. Remember, we have lactose. We'll need lactase to break the bond between the glucose and the galactose. And we'll need sucrase to break the bond between the glucose and fructose in sucrose. So now we're done. We're down to monosaccharides right now. We have mostly glucose, a little bit of galactose, a little bit of fructose, depending on the composition of our meal. So maltase, isomaltase, lactase, and sucrase are all additional enzymes that we need in addition to the pancreatic alpha amylase. All right, so now we're down to monosaccharides. Here are our enterocytes, and there are the microvilli. Now these sugars are too polar to go across the membrane, so we will need aid of a transporter that's shown right here. So these transporters, this is GLUT2, so GLUT just means glucose transporter, and there are lots of different glucose transporters in different cells in the body. So this is called GLUT2. So even though it's a glucose transporter, it's able to transport both glucose and galactose into the cell. And GLUT5, again, it's a glucose transporter, but it's able to transport fructose. So now we have glucose, galactose, and fructose are inside the enterocytes, but then they leave again. Through other transporters on the basolateral part of the membrane, the glucose and the galactose leave, and the fructose leave, and now they enter the circulation, and they will connect to the portal vein, and the portal vein travels directly to the liver. So all of these monosaccharides are going to go straight to the liver. And that is shown here in this slide that I showed you the other day, where we have the small intestine here, and then these show the blood vessels that drain the small intestine, and they all converge upon the portal vein right here, which is shown in blue, and then that delivers all of those monosaccharides to the liver. So then the liver decides what to do with them. So the liver can use any of these sugars just as an energy source just for its own self. It can store them or can pass them along to the general circulation. So the liver takes about a third of the glucose produced by digestion. And once the glucose gets inside the liver uses the glucose to make something called glycogen, and that's a compound that looks very much like amylopectin. So the glucoses are strung together to make glycogen, and the glycogen is stored in the liver for later, so during maybe a fasting situation. And then the rest of it is passed on to the blood. And the glucose can be taken up by muscle or adipose tissue or the brain. The brain needs quite a lot of glucose to function properly. The liver takes up nearly all or nearly all of the fructose. So very little fructose is passed on to the rest of the body tissue. So all of the fructose that enters the liver through the portal vein stays in the liver. So this is illustrated here. So again, we've talked in detail how the dietary carbohydrates are completely broken down into monosaccharides, and then they're absorbed by the cells that line the small intestine. They travel to the liver via the portal vein, and once the glucose gets there, the liver will take these glucoses and make glycogen. So you can see that looks a lot like amylopectin. The free glucose can then leave the liver, and the brain and other body cells will use the glucose. Some of it will go to the muscle, and the muscle is able to take this glucose and make glycogen as well. So now we have glycogen. We have a storage form of glucose in two places. We have it in the liver and we have it in the muscle. So I wanted to show you what glycogen looks like. You might notice that I actually used the same picture as I did for amylopectin. So again, 50,000 glucoses, all alphas. They all come from glucose that were derived from the diet, and the liver has enzymes that are able to synthesize, take those glucoses as raw materials and synthesize glycogen, and that happens both in the liver and in the muscle. So the hormonal response is the liver takes up about a third of the glucose and then passes along 2/3 of it to the bloodstream. That's going to alert the pancreas to secrete the hormone insulin. So the pancreas is sensitive to blood glucose concentrations. So after a meal, the pancreas will secrete insulin. Insulin actually doesn't respond to fructose, but we don't have that much fructose there anyway because usually the liver takes it almost all of it up. So the insulin ensures that everything that I just talked about takes place. So insulin will act on the muscle and the adipose tissue and cause those tissues to take up glucose. So if you were diabetic and didn't have insulin, the glucose would just stay in the blood. It would never get inside the muscle and the adipose tissues. The other thing that insulin does is it stimulates the liver to synthesize glycogen. So when the glucose comes up to the liver from the portal vein, insulin needs to be there to stimulate the liver to make glycogen. So what happens is after you eat, your blood glucose concentration goes up as the glucose enters the blood, and then insulin is secreted, and then your blood glucose goes back down again because when insulin is secreted, insulin stimulates the cells to take up the glucose. So then the blood glucose concentration falls after that. So basically what I've said in the last slide is illustrated here. So again, you eat something and then your blood glucose concentration goes up. So in this figure, these green hexagons are glucoses. These little purple balls are meant to be insulin. So the pancreas secretes insulin in response to your blood glucose concentration going up. And insulin is needed so that other body tissues will take up that glucose. And again, glucose is used by the liver to make glycogen and by the muscle to make glycogen. Now, if you eat a lot of glucose, more than you're burning off, the glucose can be converted to fat, and the insulin assists in that process as well. So if we go back to our dietary guidelines, the recommended dietary allowance is 130 grams per day of carbohydrate. That should give you enough glucose to supply your brain. The acceptable macronutrient distribution range, or the AMDR, is 45 to 65% of calories. So there's a pretty big range there. It depends on how much protein and fat are also in your diet. Now, what we should do, though, is all carbohydrates are not created equal. We should focus on fiber-rich carbohydrate foods. So, again, this is from our textbook. So 130 grams per day. So all carbohydrates are not created equal. So fruits and vegetables have carbohydrates, but they're also packaged with fiber and vitamins. There are lots of, you might have heard the term uh, uh, empty calories. So if you have something like a soda or a tea or Gatorade or something like that, those contain added sugars. And those should be less than 25% of the total energy intake. So if we look at this example from our textbook, we have something that looks like Fruit Loops and something that looks like oats right here. Both of them have the same carbohydrate, but if you have total carbohydrate here is 26 grams and here is about 27. So you'd think, oh, these are both adequate sources of carbohydrate. But this source has only 1 gram of fiber, and this one has 4. This has a lot of refined carbohydrates, where this natural whole grain oats is packaged with fiber. So one of the consequences of that is that when the insulin is secreted in response to a meal, you'll get a very large spike from something like Fruit Loops because those are digested very quickly, and the blood sugar goes up very quickly. Because the oats are packaged with fiber, the digestion is not as rapid, and that means the release of glucose to the blood is also not as rapid. So the insulin response is slower and more sustained. So here are my references. Thank you for your attention, and I will see you in class.